Good afternoon, everyone. Thank you for attending to this session. I think it's time for us to start. It's a very pleasure for me to co-moderate here. I have here Victor Parro. He's a HISAC member. He's also from the Astro-Biology Institute from Madrid. He's going to be co-moderator with me. And this session will be about the origin of life and limits of life. And I would pass the word to Victor now.
Okay, thank you. Well, I'm really glad to introduce you a great panel of scientists that I think represent very well the astrobiology in Europe. I think they all have now are leading or have led several experiments in the ISS, so they really know what they are talking about and how tough, what are the challenges of the space station and how to do experiment to take advantage for astrobiology experiments. So we will start with Jean-Pierre de Vera from the German Aerospace Center, the DLR. So he has led several edition of the BioMex experiment and now the bioscience, and he's very involved also in several missions to Mars, like he's part of the science team of some instrument, ExoMars, MSL, Mars 2020, and he's very concerned about searching for biomarkers and life. So he's going to show us how bioscience can help us to do that. Okay, so please.
Thank you, Victor, for this nice introduction. Particularly, so a lot you have mentioned, but I have also to emphasize this is actually Iberic power here, because you are also Portugal and Spain are here present, and myself, I had ancestors from Spain, so it's starting with Iberic power now. But besides of that, I would like to show you, because I have just time for 10 minutes, a little bit what is bioscience, and particularly what is the meaning of this experiment, because it's an experiment foreseen for the ISS. We are not sure if we realize it on the ISS or another platform. We try to realize it on the ISS still, and it is related to these bodies you can see, Mars and the icy moons Enceladus, so these Saturnian moon Enceladus and the Jovian moon Europa. Particularly, this is a focus. And we have a big team, so all the names are mentioned here. Just to emphasize that my co-PI is Michael Baquet. He's there in the back. And if you have more questions, please also ask both of us. Maybe that there are a number of questions. Well, we are focusing on preparing future space missions, particularly also in the range of life detection. And this is in this context. But besides of that, a number of, just in astrobiological range, outcome work was in the past in the BioMex project, the previous experiment we have done, and we see that the potential has also a big potential to the society-relevant topics. And so just keep your eyes open at the end of this talk why this is also relevant to other topics. Here is just the scientific objectives. We are focusing on one hand on the limits of life, because we are studying organisms, also which is cross-related to habitability, which means some of the organisms, if they are able to survive simulated conditions on the ISS, which are approaching Mars and Enceladus and Europa, this is a big potential also to classify these planetary bodies as habitable. On the other hand, we have a big focus also on stability of biomolecules, which means also checking about microfossils, can we detect them, can we determine it, what is about organics and biomolecules as detectable biosignatures. And we are all doing different kind of phases. We are selecting samples in the field and also trying to find out the detection under simulated conditions on ground during the environmental conditions and in space before and afterwards, because we have no facility that's available to do it in parallel. But there's another experiment you will see later also will be presented where it is also feasible to do in situ science. So to maximize all scientific results, BioSign comprises all project data generated during planetary analog field site studies. This is starting next year, where we will have also some new fresh material from different kind of field sites, including Antarctica, planetary simulation, and at the end, lab experiments. The milestones are interconnected to the objectives. We try to find out about the habitability of Mars and the icy moons. On the other hand, detecting biosignatures, which are also related not only for Mars, but also the icy moons. What is about the characterization of biomolecules, also in cross relation to chemistry and prebiotic chemistry. We will see if we can do it really. It's tricky. I talked to the expert just the last days, who is in our team. It's not easy to do this, but we try. Yeah, and check also the instruments which are foreseen for the future missions, if this is feasible, and if our tested molecules are also detectable. It has relations to all the missions we have actually in the loop or are still present. Direct reference to ExoMars and Mars 2020. We have also even included in our team some co-eyes of these missions. Direct reference to biosignature detection in icy moons in terrestrial environments. This is missing because there are also some co-eyes in some missions which are actually going as JUICE and also Europa Clipper. We go, do you hear me still? Okay. We have a strategy for just particularly when I'm emphasizing on live detection, also to test some of the instruments in the field to see how we can detect, what do we detect, and afterwards doing simulation experiments in the labs with the facilities which are available. And afterwards, then all these samples, and this is also kind of selection regimes, which were really which fit for the experiments, will be exposed in space. Maybe in future we have also in the loop the moon. We will have a look on what is going on at ESA or NASA, if there is a potential also to use the platforms on the moon. At the end, we will feed our databases, which are available with all the information about the molecules we have, also about the stability, about the protocols, about meta data according to the environmental conditions, cold, different temperature regimes, humidity, and so on. So all the parameters which are necessary. I have another slide where you can see also the metadata. Humidity is mentioned, radiation, pressure, but humidity is much more in relation to mass. So radiation, pressure, pH, salinity, all these metadata has to be also taken into account because the molecules could be have another fingerprint as it is normally under normal Earth conditions. It has also a big connection to Mars sample return, particularly because we get samples back from space. We get it from the ISS. We know how to handle these samples. We know that the samples are also transported in sealed compartments, which are, on one hand, if it is related to Mars under Martian atmosphere conditions, on the other hand, if they are evacuated under evacuated conditions. This can be transferred from one lab to another and to handle the samples under these conditions and also to see the detection, if there's a changing of the detection during this kind of simulated Mars conditions or icy moon conditions, or if there's no difference to what is under the lab conditions. So we have exercised this in the BioMax project. We will do it in the same way in BioSign. So we have also still available some databases about these fingerprints. On one hand, minerals, which we will use also on the other hand, on biomolecules and on the organisms themselves, because we are also using organisms and also viruses and so on. This is the list of samples we will use. Just to let you know that there are also a number of people here present. I just would like to mention that there is a cyanobacteria and purple sulfur bacteria community, which is included particularly in the relation to Mars and also habitability of Mars. Some lichens are used, so a number of organisms. But what is also new in this experiment is icy moon-related samples are here. Karen, are you here? Is not here, but Frank is here, who is working with her together. Just shake hand. Then there is also cyanobacteria. This is Daniela. Where are you? You are here. I have another look on others. Methanogenic archaea. This is not present here actually, but then we have also a number of biomolecules. So Michael, I mentioned that just before. Who is here from the Bioscience project? Just raise your hand. Okay. Some are just missing. There's Andreas also at the back, because he has also another experiment just before us to be done. Yeah, and we have also microfossils. The people were not able to join here, but this is also a new kind of samples we are doing. So when the call of ESA was, there were different kind of facilities at that time, but actually ESA has decided to make a new one. This is just an example, because this was when we started some studies, but the final facility, I cannot tell you actually, it's just in preparation. But approximately like this, where we have some different kind of hardware where we can find then also, where we can insert the samples. And then we have different kind of methodologies. I have to come to an end. I know that time is pressing. Just to have an idea about the sample preparation, how it will be integrated and then exposed to space. And just to let you know that the Bioscience project is about more than 16 institutes actually, because some joined just recently, distributed in 14 countries and on 4 continents, approximately 50 scientists. This is often varying because also of the students which are also joining. So this is a big community, and yeah, I have to admit that this is really a big project, but I'm happy about this because so many countries, and it was also in the Biomax project, and still new countries joined us to work on it. And now this important slide to let you know the potential of what is coming out of such kind of project. Yeah. We have some connections to the health community, particularly because we are using an organism consortium, like for example kombucha, which could trigger the immune system. We have done some studies under the Biomax project and try now to make a replicate to find out what other potentials are there. This community is stable, is really the key organisms are even resistant to space conditions, and this is important to know how we can use it. Some of them are also producing cellulose, which is very well known, where you can also do some products with that. Another health issue, you know about the pandemic situation. We approach it with phages infecting some bacteria, and to see what is going on with these kind of interconnection with these organisms, which are exposed and before and afterwards, and see also about the genomic changes of these organisms. Agriculture, climate change, this is also interconnected to some of the cyanobacteria we are using, because some of them are nitrogen fixations, fertilizers, and can be used also in relation to the first session I mentioned just before, to plants. Yeah? The enrichment of nitrogen on Mars is also an important issue, particularly as you have some nitrogen in the atmosphere available, and this can be used because, and on the other hand, oxygen production, which is closely related also oxygen to biofuel. It can be a biofuel, even the organics can be a biofuel. And the biological life supporting system, we are also thinking about the human to be transferred into space. So this is all interconnected, and also the ISRO biomining, for example, biomineral combination resource gain. And biomonitoring, biosignatures investigation could lead to faster detection of biological systems in the environment. The tricorder principle of, you know, Star Trek. Sorry. And with that, just an outlook. What does it mean to have such a big project? A lot of publications, in also a number of books and articles. We had at the Biomax project, the previous experiment, about now more than 60 publications. And with Biosign, we try to reach the same level, maybe more, we hope. And with that, I would like to let you just have an inspiration, a slide. Yeah, we cover all the aspects of astrobiology field studies, lab work, space, maybe in one day on the moon, to support future exploration missions, including also exoplanets, yeah, because think about the purple bacteria. With that, I would like to finish.
Yeah, okay. We have time for just one quick question. I prefer that to make questions after the talk, so if someone... No question? I have one quick one. One of your experiments is testing the stability of biomolecules. What do you think is the most critical parameter and also the most relevant in the case for searching for life on Mars?
I would always say...
Time is
absolutely. I would always say radiation. We had the most impact even in Biomax experiment on radiation, which is really affecting a lot the molecules. Some molecules, not all. And it depends also because we have also the minerals. I have not talked too much on that. Some of the organisms are also embedded or on the surface, but it's surrounded by minerals. And there's another interaction, sometimes bad, sometimes good. Some are protecting, others not. It depends on the interaction with the radiation. Yeah, there can be chain reactions, but some are not affecting. And by these experiments, we find out which molecules are really stable. But the majority of the molecules are very sensitive to radiation.
Thank you. Okay, next one is Daniela Billy, professor of biology in the University of Rome, Tor Vergata. She's an expert in the field of astrobiology, but with model organisms, which is cyanobacteria, Extremophiles cyanobacteria, and I'm pretty sure that she's going to show us how cyanobacteria can help us in biomex experiments and bioscience. Okay, thank you.
Okay, thank you. Okay, yeah. Yes, I will give a very quick overview about what has been done in the past using desert cyanobacteria, and what we are doing currently, and what we would like to do in the future. And of course, every new opportunity is a challenge to make the most, to get the greatest results we can from the experiment. And as Victor said, I work mainly, actually exclusively, on cyanobacteria, on desert cyanobacteria that on Earth colonize extreme environments like hot and cold desert that are considered Mars analogs. For this, there's been, since the very beginning, considered of great interest to astrobiology. So since the time Imre Friedman and his wife Rosalie Ocampo discovered this community in the dry desert of Antarctica, in the dry valleys, it was 1976, so it was the time of the Viking mission that failed to give us proofs of the existence of life on Mars. And since then, this cyanobacteria, actually cyanobacteria of the genus Crocosidiopsis, which I will not name anymore, but the genus is Crocosidiopsis. I will just say cyanobacteria. That's been used in really a lot of experiments that has been provided by ESA, and from my side financed by the Italian Space Agency. And we started with the SON experiment. All the experiment that has been performed outside the ISS have included cyanobacteria of the genus Crocosidiopsis. From my side, I started to get involved in this experiment with the exposure to space mission. As Jean-Pierre said, we are all looking for the biosign to come, but these are passive experiments that are really very important and interesting, because once that you have the sample back, you can study the stability and the detectability of molecules that has been exposed in the dried state to Mars-like condition or space conditions, so help us to search for life elsewhere. But as a community, I think that I can say that we are all looking for active experiment to be performed in CubeSat. We are looking to the space ride or the moon surface, or the moon surface, and these are, of course, are very challenges, because sample return may be no guarantee, and so you need to have in situ analysis, so it means that you have to perform active experiment on hydrated organisms, and this is very challenging. And of course, we know a lot from the past, so I will go here very quick, but desert cyanobacteria has been used in the Biomex experiment, so we figure out that protection against mainly UV radiation was provided by regoliths that are shielding the cells, and likely UV radiation are not so penetrating, so little is enough to give protection. And I also want to stress that the ionizing dose outside the ISS for 1.5 years is really not that much for this kind of organism. And also we had a lot of data also from ground simulation, like in the StarLife campaign. We irradiated the dried sample or hydrated sample with gamma rays, and we figure out, of course, that there is a very high resistance when cells are dried because there are no ions. And I want to mention here that we can detect DNA at a very high doses of ionizing radiation, where actually cells are dead, but you can still amplify DNA, and the dose corresponds to 30 millions of years under 2 meters from the Mars surface in a dried state, so it's a kind of contribute to the Rosalind Franklin mission. And also we did with Parro, with Victor Parro, also we used cyanobacteria cells that were exposed to UV and gamma rays, and we used this sign of life detector, and we came up with the conclusion that signal was still detectable after high doses of ionizing radiation, the same doses that I showed you before, so 113 kilograms of gamma rays, whereas UV doses are really very damaging, and the dose that correspond to a few hours on the Mars surface are already damaging the possibility to detect molecules that are, let's say, present in the cell and detected with an antibody approach. Also, in the frame of a project that has been funded by the Italian Space Agency, we have been working in the project LIFE. And in this context, we investigated desert cyanobacteria that were able to perform oxygenic photosynthesis beyond the visible life. These are very important, as Jean-Pierre said, also to contribute to the search for life, not only in the solar system, but in planet around stars that are emitting not in the visible light, but in the far red. So this cyanobacteria can produce a chlorophyll F that absorbs outside the visible. So this is a suitable model organism to perform a new experiment also in space, mimicking maybe exoplanetary condition. And so if we want to move towards active experiment, we have to address a lot of things. Maybe we want to integrate the cell in the dried state. We know that desert cyanobacteria can stay in this state for years, then radiate in orbit, and then decide what kind of in situ monitoring we are doing. This is the challenging part of doing this. In the frame of the Astro-bio project that is just started and is funded by the Italian Space Agency with other Italian university and also the Kaiser Italia, we are developing an experimental unit and to find the and find, sorry, I made a mistake. Back, okay, here. And to find the proper approach to monitoring cell viability in remote, and so we have also to deal with the autofluorescence of pigments, so it is not so easy to find the proper approach, but we are working on that. And also we are working on the Extremon experiment that is part of the ESA pool of science activities on the moon, and in this case we expose dried cyanobacteria to ionizing radiation on Earth. So in order to induce DNA damage, we will radiate this in space to allow DNA repair and have the sample back fixed and do genomic sequencing to see if the repair of the DNA damage in microgravity, in altered microgravity will be efficient, and we will, of course, this has to be, and we will do this on the ground using a clinostat to get ready for this future experiment. But what we are doing right now is to get ready for the BioRider experiment, Cyanotech Rider, and the approach is the same. We will expose dried cyanobacteria to ionizing radiation, radiate in orbit, and when it comes back to Earth after 2 to 3 months under microgravity, because this is the Space Rider mission, we will do again the genome sequencing, and we test, and we will see for real if DNA repair will be efficient in microgravity compared to what we already know as part of the experiment that we did after the Biomax mission, where we recovered the cyanobacteria after 1.5 years in the dried state around the Earth, so outside the ISS. We recovered the strain, and we sequenced the genome DNA after recovery on Earth, and we found out that there was no increase of genome variants. But this, of course, the DNA repair was under Earth conditions, so it is important to see what happens in altered microgravity or in the absence of gravity, according to the platform that we will be using in the near future with BioRider, or maybe in a more long distant future on the moon. And we are also working developing a biosensor using Crocosidiopsis engineerized in order that we will monitor in the luminescence after the occurrence of DNA damage and activation of the SOS DNA damage, and this will get us ready to perform some experiment on board the CubeSat. So it is very challenging, of course, to use this cyanobacteria to know the limits of life as we know it, to see a fundamental biological issue under non-Earth condition. But this kind of astrobiology experiments are not just standing alone, but there are a lot of, implication. For instance, we can use cyanobacteria for life support, for synthetic biology, to find out how they survive this high doses of ionizing radiation and find new countermeasure for humans, for instance, or if it is not for humans, at least it is we could apply these strategies to organisms that are used in life support system. So as a member of the topical team that I coordinated with Andrea Alceser, we wrote up all this indication and what we wanted to do. And so our desire is to have a new platform and to go far from the low Earth orbit, to have active experiment, to have higher dose of ionizing radiation, and to have also miniaturized experimental unit to accommodate this experiment. And with this, I'm done.
Thank you, Daniela.
Sorry, I don't know why there was a slide missing on the BOSS experiment, bioorganism surfing space. I don't know why.
It's okay. Some question? Any question? Come on, someone. No? So I have one. Have you found any changes in the pigmentation of the cyanobacteria after exposition, or not only chlorophyll production, but I mean any other pigment that can be triggered for protecting against radiation?
So in the passive experiment, of course, we cannot see anything because they are dried. So I can just say that the layers that are exposed are, of course, bleached. So far, it's just the damage of pigments that are reached by radiation. If there is the biofilm, is the top layer. If it is a mixture with regoliths or maybe cells that were not protected by the grains of the regoliths, but all these are passive experiments. So passive on dried samples, so there is no answer, no? There is no metabolic answer, and so a protection put in place, no?
Perhaps when you have this monitoring system
then it would be much better and to see how cell really responds to space condition, because now they just accumulate damage, and then we can see how far they can repair the damage that has been accumulated while in space.
Thank you.
I have one question. It's just like the Alcyon is. You said that was in a CubeSat. Is going to be in a CubeSat?
The CubeSat is not attached to this proposal that is funded from the European Community, but it is in preparation, and there will be a lab on a chip done by Mara Mirasoli and Augusto Nascetti from the Italian Space Engineering, La Sapienza Engineering Department.
And a more general question, because you saw a lot of nice experiments that you're planning on your pipeline. Let's put it. Is there, in your scientifically speaking, you are thinking on some kind of overlapping or follow up on the results, or they are totally independent?
No, they are all mixed.
All mixed.
These experiments are all mixed.
Okay, thanks.
They are different, but they require more or less the same knowledge to go ahead.
Thank you.
Thank you. So next speaker is Adrienne Kisch. She's Associate Professor of Microbiology in the National Museum of Natural History in Paris. She loves extremophiles, and overall she's an astrobiologist, and she is a part of the science team of the ExoCube experiment, and she's going to tell us some details about how microbiology can contribute to the experiment. Okay, thank you.
Well, thank you guys so much for the invitation to come and present, and my talk follows right on from Jean-Pierre and Daniela, so this is perfect, because what we're going to talk about right now is exactly how do we make this transition from passive experiments to active. And so to give you a bit of the history, we're going to take the example from ExoCube, but to put that into context, just a reminder, as Jean-Pierre was showing us, astrobiology is science from the ground up. So the way that we pipeline this is that we look at extreme conditions on Earth, we do culture in the labs, we study these organisms that live in environments that are analogs to space. Then you try to simulate the space environments in the lab, and once you have all that data, then when you need to put it all together, you can access the space environment with all that information and really utilize the space platforms most effectively, using everything you can to simulate first, and then using the space platform when you need to mix all those conditions together. Now, as you've been seeing up till now, things have been done in this way intentionally. This is not an accident. We haven't come across this, it's a design. We want to do the science this way, bottom up. And I've been referencing a lot of these documents from topical teams, from the communities that have been doing this, from the astrobiology community, so that you can check out these documents. I highly encourage you to do so, because you have all the recommendations from the scientific communities there. So traditionally, how the science has been done on passive experiments is that you culture the microorganisms, dry them, and then bring them to space on the platform, expose them dried, bring them back down to Earth, rehydrate them, and then do all the molecular analysis. And this has been very successful. But this requires sample return, and we're now going into the post-ISS era. We want to go beyond the ISS, use different platforms. We want to take a look at CubeSats, at Lunar Gateway, going on to Moon, Mars in different ways. And that's going to require in situ science. So in situ science, the advantage of this, why we want to invest into this, is that because, first of all, it's automated. It means you don't need astronaut time. You don't need astronauts to have to do that, which is great for us in the areas where we're going to have this now as gap before we have crewed missions again. And it's also compatible with beyond low Earth orbit. And so we can start looking at autonomy. And this is something that has been identified in previous ESA roadmap activities, looking that we have to have access to different space environments for in situ measurements. And in situ measurements have been noted by the communities as well. Look at the latest road mapping exercise. In situ measurements were recommended by the astrobiology community for all these different scientific areas and major scientific questions. And they've been identified for all those different platforms and destinations. So this is really cross-cutting. The other thing that to make it even more interesting is to go from in situ to adding microfluidics. So again, from these papers, we can see that microfluidics has been identified as a key in situ measurement technology. But microfluidics is not just for astrobiology. While the astrobiology community has, again, identified it for many, many of their major questions and key issues for looking for life in the environment, origin of life, biosignatures, and different kinds of destinations and platforms, it's also been identified by other biology communities, including for human health, for tissue chip, for things like that. And, well, for planetary protection studies. So this is really cross-cutting through a number of ESA's objectives. So this brings us to where we are now with ExaCube. So ExaCube, the PI is Andreas Assesser, who is here in the room. And he is bringing together this platform that's going to test all of these different organisms. It's very similar to what you've seen with BioSign and some of the cyanobacterial experiments that Daniela was just mentioning, looking at different biomarkers, molecules addressing areas of origins of life, and also survival of microorganisms. The difference is this is going to be active and in situ experiments. So while we're addressing these questions, which will help us address key astrobiology issues, but as well help us to address planetary protection and other key issues that ESA is wanting to address, it's also international. So as you've seen from the other experiments, we don't ever do this alone, and this is a place where Europe has a strong advantage, is because we do international collaborations between the different countries under large groups. And if you look at just at the interaction that Andreas and I are working with, and you'll see that my hair color is very much a subject of change. So the samples that we're providing are halophiles. So we're looking at salt environments. Why salt? Salts you can find, regular table salt, NaCl, in different planetary conditions. And so if we want to look at biosignatures for life, it may be contained within salt crystals. And salt crystals have been shown on ground-based experiments to provide a level of UV protection, so we can actually protect the biosignature on the inside. Those are some of the reasons why we want to investigate it. It's also based from the ground-up approach. So these are organisms that have been highly studied at all the levels, from the cell to the omics, in ground, in simulations with all the different kinds of radiation that we'd have in space individually. Now we need to mix it all together and put it into space. So we're really going to try to access this space platform and get the most that we can out of it. It's also inspiring. So these are some of these people are here in the room, including David Burr, who's been a huge contributor with Andreas to the development of this project. And for every one of these early career researchers, we have a 20-fold number of applicants who want to come work with us. This is inspiring. People want these kinds of jobs. We have trainees. So we have a really engaged generation that we want to bring on board for this kind of science. So the science is going to be put up on Bartolomeo platform on the outside the Columbus module. So it's not going to be inside ISS, it's going to be outside the ISS. As you can see, ExaCube is separated into two components. ExaCube Chem, which is going to be looking at biosignatures similar to BioSign, and ExaCube Bio, which is going to be for the microorganisms. So we're not going alone, we come with friends. It's the other experiments that are coming on board, and this is going to be what used to be Expo, and it's now Exo-bio. OreoCube and Ice Cold are accompanying as well, and this is going to be on the Exo-bio 1 that should be going up in 2026. So to go into a bit more detail, ExaCube is separated into two components, both in situ. So this is all going to be in situ science happening on board. So we're going to get the data in flight instead of returning the samples. So for ExaCube Chem, it means that we're looking at an infrared spectrometer. that's going to be looking at cell membranes, different molecules, et cetera, that have been dried onto underneath the magnesium fluoride window, which allows for the radiation to come through. Now, I spent a little bit more time looking at Extremophiles because this is our microfluidic system. So we're going to have a microfluidic system. The samples are going to go up dried. They're going to be rehydrated in orbit with fluorescent markers for metabolic activity. So we're going to be able to see if the cells are alive, are they proliferating with optical density, and are they metabolically active with fluorescent markers. So you can see this is several steps ahead from where we started, which means it's a major challenge. Any great step forward is going to mean a lot of challenge in the hardware, but we think it's a challenge worth investing in. So we look a little bit further at this. Where do we've got so far? So even just with the pre-flight, we now have a new set of biomarkers for fluorescent markers that can be used and are compatible with spaceflight conditions. And we're able to make that applicable with fluidic systems. Not to say this is all easy. It's not, because when you do fluidics, you basically have to take an entire microbiology lab and stick it into a cardboard box. In our case, it's actually all of this equipment that has to go onto something the size of that, which makes it a major challenge. So this is what I want to end the last few minutes of my talk and talk about. So how does Extremophiles work? In general, I want you to show we have this system. So we have the samples are going to go here underneath the window, and there's going to be a shutter. When we get into space, we can open the shutter. So in the case of just the samples that I'm going to be contributing and my team is going to be working with, it's Halobacterium salinarum. It's going to be dried inside salt crystals, inside fluid inclusions of salt crystals, and they'll be sent up dried inside their salt crystals into space. Once we get into space, we open the shutter, let them be exposed to radiation, and then we can inject their growth medium and the fluorophore after we're finished exposing them to radiation and close the shutter. So at that point, we apply a piston and essentially inject it, rehydrate the samples, let them grow. We can do optical density measurements to see are the cells proliferating, are they dividing, and we can use the LED for fluorescence to see the metabolic markers. So are they dividing? Is that real growth? And are they active? Then we can inject a chemical fixative. So then we can fix the cells, try to bring them back. If possible, this is almost bonus if we can get the samples back, because we still have the luxury with ISS of sample return, which we won't afterwards. And we can do things like electron microscopy. So what are the lessons that we learned that we should show for paying it forward to the next generation of technologies? First of all, there is a real driver to invest in some of these cross-cutting technologies. It's a lot of investment up front to get this hardware, but this is hardware that can be applied to other things. And I really want to thank ESA for opening up this internship that's going to start by cataloging all of the past experiments, what's been done, lessons learned, so that we can take that as a roadmap for moving forward to new technologies. It also needs to be linked to the ground-based work and the future platforms. As I mentioned, fluidics is everything from human to molecular to microbial. So this is a key technology we want to invest in and can be used on Earth for extreme environments. All those sort of studies can be applied in space. And so we want it for both of those platforms, even when it's not related to spaceflight. It's also been identified as a key technology as we go into Lunar Gateway. And that has also been contributed right now into the METIS facility design team. So this is looking at how we're going to use Lunar Gateway for science that's life sciences and material sciences mixed together. And microfluidics was identified as a key component. This is a group led by Nicole Kaplan at ESA. So we also know that we need, from this experience, we need to have a common language between science and engineers. This is engineering like we haven't done before, an interaction between science and engineering that's new and it's difficult. So what things that we learned, an example from METIS is when we had Nicole Kaplan integrate the group, we had Beth Lomax, who's there from the engineering side, that help us translate. This is what a scientist says, this is what an engineer hears. And so that's a good experience to try to get us on a common language so that we can build these new technologies. So one thing that could help would be things like having ESA have workshops before you propose something to help you design requirements. Have engineers in the room saying, if you tell me this is what I hear, to help you design requirements. And even if we're using private space contractors, that would help as well. More interaction between science and engineering at the start of the project. Have the engineers come to the science lab, see how it works with all of our equipment. Have us go to their engineering facility to understand how they're actually going to build it. And when we have expertise and things like fluidics that are being developed across different agencies, as scientists, we already know how to contact our colleagues, and we can talk directly with them across borders. Having the engineering teams be able to talk to each other would also help us advance and develop key strategic technologies. We also want to do interdisciplinary science, and again, from experience from METIS, we found that when we had life sciences and physical sciences in the same room, we found ways that we could do experiments simultaneously, which is very interesting. And workshops like this gives us chances to interact, but it means that ground control facilities are key because it's very, very complicated to do ground controls with large experiments. So facilities like MUSC are a good start, but we need systematic access to these ground control facilities. So it's all that together. This is going to help us embrace this new era of technologies beyond ISS and help us with active experiments. So thank you very much.
Questions? No question? Well, I have...
Natalie?
How does the mixing or the homogeneous distribution of your stride cells under microgravity or lunar conditions, or how do you think about that?
Yeah, we're about to find out. It's one of those things that's very, very hard to simulate on the ground, and so the part of the return from this experiment is going to be finding out exactly that and how well it mixes so that the next iterations, the next generations can take advantage of that. But that is a key issue, and this is one of those areas where we're just going to have to see.
Hi, Adrian. Thank you for your talk. I'm just wondering, the Astro-bio one, if you will measure the metabolic, right? And you will have dosimetry as well. Radiation dosimeter is going to be active or passive?
So we're going to have different kinds of dosimetry on it as well. So we will be able to have all the measurements. So we want to have total radiation, and because we want to be able to shut off the different experiments at different times, because not all the samples want the same radiation dose. So we're going to have both kinds of measurements so that we can treat each sample separately. We can talk more about the exact details after.
Okay, thank you, Adrian. So next speaker is Boris Spils. I hope I have pronounced well. Spils. Okay. He's from the Laboratory of Evolutionary Genetics and Ecology from the University of Namur in Belgium. Perfect. So he's been involved in many projects in the ISS too, and now we move from microbiology to small animals, rotifers, who is an expert. So he will talk us about the resistance of these amazing organisms to space conditions. Okay, thank you.
So today I'm going to present you the Rotifer in Space project that is organized by 3 different groups in Belgium in close collaboration with the European Space Agency. But maybe first introduction to the rotifers. If you are interested to find your own tiny polyextremophile friend, I invite you to go to your garden or just go to the street. You take a piece of moss or lichen, you back to your office, you add water and you check under your microscope. And there is 99% of chance that you will find some of these extremely interesting animals, because first you can find everywhere on Earth, so in moss and lichen, but they are present on Antarctica, and some people melt ice from 24,000 years ago, and they go back to active form. You can also find on Atacama Desert. One explanation is for the fact that they are able to resist to desiccation at any stage of their life cycle. So when they are dry, you can keep it for 1 year, you add water, they come back alive. A second important feature is their capacity to resist to high dose of ionizing radiation. So here you have an overview of sample. Here you have approximately 50,000 rotifers, and you can expose to proton radiation, X-ray, or iron particle. And if you want to kill them, you need to reach dose of 7,500 gray. If you compare to human, most human cells die at maximum 10 gray. And so there is a question, how these animals that were never confronted to such high dose of radiation are resisting to this radiation. So we try to investigate this question by making a link between desiccation and radiation resistance. And we were able to collect rotifer from different places of the world, including the Antarctica or the Atacama Desert. And we found what was normally described in the literature, that the rotifer are able to survive to desiccation. So here the data after 1 month of desiccation. But for some of these individuals, they were not able to dry and go back to life after. So we have here a good opportunity to check, do we have a difference in terms of radiation resistance for these desiccation sensitive species? So we test what's happening to these animals if we irradiate them to high dose, 2000 gray. So here we have the Atacama Desert sample. Here it's a laboratory. And you see that after 2000 gray, no death reported. But we have an impact on the capacity to reproduce. And here there is something interesting also for us. The Bdelloid rotifer, you only have females. There is no male. So people look for 300 years for penis or something else. There is no male. There is no mating. So with a single females, you can restart a new population. And in terms of exobiology, having an animal that is able to restart a new population, that's also something interesting to have. But so if you want to sterilize 50% of your population, you need to reach 1000 gray of radiation. And what is happening to the desiccation-sensitive organism? So we observe that when irradiate hydrated, the survival rate was still high, but for example, for these species, we see a significant impact of low dose of radiation. So when I'm speaking about low dose, it's 25 gray, so it's still killing a human. But we see that these animals seem to be more sensitive to radiation in terms of capacity to reproduce. Okay, but what is happening to these animals? A first hypothesis might be that the radiation breaks your DNA, and potentially in these species you can repair your DNA, but here not. So we perform pulse field gel electrophoresis, where we check how the genome is completely broken up after irradiation. So you can imagine that, so in human cell, one DNA double strand breaks lead you for a cellular suicide. Here we have the genome that is broken into hundreds of pieces. And post-irradiation, you see that the small DNA fragment reduce, and we have a recovery, a large DNA fragment supporting the presence of active DNA repair post-radiation. So what's happening for the desiccation-sensitive species? Okay, that's in fact exactly the same. So trying to define the radioresistance by the presence or absence of a DNA repair system is not sufficient, because here they are still able to repair their DNA, but they are sterilized by the radiation. So the step further is to try to understand which molecular pathways are involved in this desiccation and radiation resistance. We perform a wide-scale transcriptomic analysis where we try to evaluate the transcriptome of hydrated rotifer, what's happening when they are just dry, what after radiation, what after different point of radiation. We try to compare the impact of low or high LED radiation, and also what's happening when they are hydrated and irradiated to extract the contribution of the desiccation process. And we were able to find differentially expressed genes in all of the conditions. And as it was already mentioned yesterday by Marco Durante, we were able to find effectively some pathways are common to desiccation and radiation. But you have also some difference, and you can find difference from low and high LET radiation. So I will not go into detail for this figure, but in terms of DNA damage, we can look for the DNA repair damage pathway. And what we report is the fact that apparently the pathway that is the most differentially expressed in terms of DNA repair, DNA double strand breaks repair, is the non-homologous end joining pathway that is known to be non-faithful. And it was quite contradictory to other results we get, where we show that the genome can be broken, but also effectively repaired. The chromosome can be repaired. But the homologous recombination, okay, we have some genes that are differentially expressed, but if you compare this with this, you clearly see that there is more impact on the NHEG. But here we are working on whole animals, and so we are at the next step where we try to investigate the RNA localization at the cell level stage, and we now describe that the NHEG is expressed in all the cells of the animals, and the homologous recombination is only occurring at the reproductive level. And that's also logical for the production of your offspring and conservation. So we use these animals to investigate how the microgravity can affect these animals. And so 4 years ago, we have the exposition of different bags with rotifers to 12 days of microgravity and, for me, what is a low dose of radiation for rotifers. So for astronauts, it's 100 times higher. But, okay, what is happening to the transcriptome of these species? So just to the take-home message, we have differentially expressed genes, but as you can observe, if you are familiar with transcriptomic analysis, we have no big log-fold change. The DNA repair genes are in pink, and you only have the DNA repair upregulated. There is no pink on the bottom part. And an interesting point was that the Bdelloid genome have a percentage of high level of horizontal gene transfer. So rotifers are able to capture genes from bacteria, plants, and fungi. And we report that these genes were enriched in the upregulated and downregulated genes during spaceflight. So they acquire these genes and they use to adapt to a new environment, such as the space flight. We had another experiment where we tried to desiccate the rotifer introduced in this box after irradiation, and we investigate after radiation, do we have a repair, and what are the consequences of the repair during the space flight. So we have preliminary results supporting that if you check for the genomic modification post high dose of radiation, so here you have the presence of some deletion in the flight versus the ground, and apparently in both cases you have some modification of the genome, but no more in the flight versus the ground, supporting that the DNA repair is working with the same efficiency than on Earth. And so finally, now we are preparing an exobiology experiment. So different species are selected, and we are exposing these species to a space simulator, so simulating the vacuum, the solar radiation, temperature change. This is an overview of what can look like our hardware. And the idea is to expose them for at least 3 months in space. And so when we simulate a scenario of flight, so here for 3 months of exposure to space, we observe that in the control, so we have high survival rate. If you have only temperature change and vacuum exposure, you have still 60% of survival rate. But if you had the UV radiation, all of them died. So apparently, and if you check after 1 month, we still have some survival. The UV radiation seems to be the destructor of the rotifer. So I'm alone today, but there are hundreds of people involved in this project, and so thank you to everybody for the support, participation, and I'm happy to reply to your question if you have. Thank you.
Questions?
Thank you. This repair mechanism that will take the fragmented DNA and put it back together, this is bound to be quite energy intensive. So is there a way to simultaneously look at metabolic rate? Because I suspect this is something that's really quite stressful and needs a lot of metabolism going on. And also, as a follow-up, it would be interesting if that works the same way or with the same efficiency in a nutrient-deficient environment. Mm-hmm. So can they grow back if they are starving, basically?
So first, we don't know a lot of things about these animals. So it's very difficult for me to have a complex answer to give you. But you are speaking about starvation. We know that if you dry them, if you want to have the best survival rate, it's better to have dry and starved animal. So when we rehydrate, we don't need to provide food. And this summer, we sent again rotifer to space to have an overview of their behavior under the microgravity environment. So an astronaut was checking the motility of the animals. And we use only water for the rehydration. And the rotifer were observed 2 days post-rehydration, so 2 days without food, and most of them were alive. So this is supporting that they probably have their own reserve that are using, but they don't need, or paradoxically, if you add water, food, that's not a way to have a better survival rate.
Yeah, I couldn't help but notice the similarities between Deinococcus and your rotifers and the idea of the desiccation linked to radiation. When you're looking at your molecular, do you see similar at the molecular level, the similarities between Deinococcus and the rotifers?
The problem is the fact that for Deinococcus you are in prokaryote and here you are in eukaryote, and so you have not the same way to completely repair. But that's true that in midterm research, the idea now we have is to compare the transcriptomic response of different organisms, including maybe bacteria, rotifer, or the candidate, to find a common response to this.
Yeah, you can make use of the Tanpopo data from the Deinococcus experiments they did with the transcriptome.
There is, but to my knowledge, it was not performed on the rotifer for comparative study. But again, we are not here with model animals, and so if I can go back to the transcriptomic data, in fact, I think maybe 50% or between 20 to 50% of the gene we don't know what they are doing. And so when we say, oh, this is the DNA repair, yes, it's what we know. But we have plenty of genes with no description. And if you check for the most differentially expressed genes, most of them are without any function at this time. So potentially with AlphaFold or something else, we will be able in the future to test. And we are also lacking of genetic tools to make some knockdown. And so, yeah, at this time, we can just compare with what we have.
And haven't you found any genes involved in oxidative stress resistance? Because haven't seen any of any genes that produce proteins involved in oxidative stress resistance. I don't know, whatever, oxidases or something like that. Because all you show is DNA repair.
When we made the first genome sequencing 10 years ago, one of the key findings was that the rotifer, if you compare the amount of antioxidant genes versus the key animal model, have 4 times more antioxidant than the other. So we have clearly an enrichment, but they are degenerate tetraploidy, so you have ancestrally 4 copies of the genome, so that also helps to improve the number of the genes. We clearly see that there is an impact of the antioxidant response, and we have a PhD student that really focused to find how to improve or how to use these antioxidants now for pharmaceutical research. But I think to my experience, the high resistance is a combination. It's not just one actor. Because in terms of DNA repair, one gene, ligase E, was found to be highly overexpressed during DNA repair. And you can express these ligase E to a human cells. And effectively, you observe an increase of radiation resistance. But it's still not so resistant like cyanobacteria, or just a small increase. So I think you need to have the full set of candidates to do something.
Okay.
Can I suggest to also check for autophagy genes?
Yeah.
Sorry. I was saying, I was suggesting to check also for autophagy genes, because the fact that they are more resistant to radiation in starvation makes me think that maybe autophagy could be helping recycling the damaged molecules, and so.
Yeah.
But it's just a guess.
But you are right. We see a kind of recycling candidate genes that are important indeed. Yeah. And it was in the picture, so you're correct.
We need to move on.
Thank you, Boris. Yeah. Okay. So because we are running out of time, so the last speaker is Thank you. Rosa Santo Martino. She's a PI of the Space Microbiology Group in the University of Edinburgh. Also, she's about to move to Cornell University. So we lost her here in Europe, but she will be connected with us. So she's now going to tell us about a very interesting project that tried to explode the biomining approaches that we are having here on Earth, but now on the space, and how they can help to support the human mission and make them sustainable in space. So, just.
Thank you, and also thanks for this opportunity, because as a young PI, it's really a fantastic opportunity for me to be here and tell you something about what I do. And in a way, I see what I'm going to talk about, which is microbial-based biotechnologies, as a link between this panel about astrobiology and the ones that we will have tomorrow on microbiology and synthetic biology. So go and see them as well, please. Okay, we heard a lot in these 2 days, and surely also tomorrow, that the future of human space exploration is moving farther and farther from Earth. And something that I would like to capture your attention to, though, is, of course, this. The distance, of course, is going to increase. This is kind of obvious, of course. But these numbers means that the longer the distance, the longer also the travel. And so we are also going to be, more resources are going to be needed to support long-term human space exploration. These resources include life support systems, food, water, oxygen, materials and energy. Yesterday we were talking, someone had a talk about, more than one talk about the importance of shielding. Well, we are going to need shielding material. And long-term necessities, so in the long term we are going to need pharmaceutical product, for instance, just to mention one of them. So in other words, to make deep space human exploration sustainable, we are going to need to develop technologies to obtain these resources in situ, a concept that is normally referred to as in situ resource utilization. But the good news from me, who have a background in microbiology and astrobiology, is that microorganisms can really help us on this perspective. In fact, as we know, they are widely used on Earth, including in industries such as food or drug industry. And because of that, they are going to be key on an extraterrestrial settlement. Think about the moon, Mars, or also travels in between. Which are some potential roles that they could play for us in space? Well, they could help us generating food and oxygen, recycling waste, obtaining materials, and also in some bio industries. Now, I would like to shortly mention this, like the use of microbial biotechnologies to obtain materials, which directly get into the in situ resource utilization concept. Which biotechnologies could we use for that? Well, there is the biomining biotechnology, which utilizes microbes to extract metals of interest from rock, ores, and mine waste. And it is a technology that is widely used on Earth. But of course, so there has been an increasing interest on using it in space, because of course it might make our life there easier if we can use microbes to extract elements of interest. However, as we know, the conditions in planetary bodies and generally speaking in space are quite different, and we cannot assume that a technology that works perfectly fine on Earth is going to be directly transferable to space. So we need to test that, and it's with this idea in mind that we launched the first two biomining experiments on board the space station. The first one, BioRock, was supported by ESA and was launched in 2019. And then we also had the second one called BioAsteroid, which was launched in full pandemic time in between 2020 and 2021. They have some key differences, but also a lot of similarities. So I'm going to talk about the two of them together for matter of time. The main similarities was the fact that the same bioreactor was utilized. This is the biomining reactor developed by Kaiser Italia, but also the guiding questions were basically the same. We wanted to understand first, does gravity influence the way in which the microorganisms interact with the rock surface? Because of course this is going to potentially, cause problem to the extraction of metals. And so the second question is indeed, does this affect their capacity to perform the biomining technique? Very briefly on the results, There are many publications on this, well, a few publications on this, and results, but the key one, of course, was about bio-mining. And we were very happy because the results were generally positive. We managed to see that not everyone, but some of the microorganisms that we utilize in both BioRock and BioAsteroid were able to extract rarer elements, vanadium and palladium, in all the gravity conditions that we tested. So in general, this is good news for who wants to do biomining in space, because it was indeed the first proof of concept that demonstration that space biomining could be achievable under a range of gravity conditions. But then I got very, very interested in another potential biotechnologies for space application, which is their potential use in recycling waste. Now, you might be, let's see if this is supposed to be a video. I'm not sure if it will work. It doesn't. But anyway, what you would have seen is this thing, which is a packed waste being ejected from the International Space Station, reaching Earth, basically. Now, this is the way in which solid waste is currently disposed of on the International Space Station. And it makes sense when we are orbiting Earth, because this is the most viable solution. But you can imagine how easily this becomes unviable and unsustainable if we think about going toward Moon or Mars. But luckily, of course, space agencies are aware of the problem. And biotechnologies for recycling biological waste, by which I include food waste and human waste, wastewaters, for instance, are ongoing. For instance, everywhere I give this talk, I always talk about the ESA-Melissa project, because to me it's a very clever and very nice project in which really a circular economy is under development, and each compartment is going to basically is composed of microorganisms that will recycle biological waste, so to make basically virtuous circular economy. But there is a big gap here. I don't know if it's obvious for you, but to me it was obvious. Synthetic waste is missing from this context here. For synthetic waste, I mean electronic device, clothes, consumables, a whole bunch of synthetic waste, which if you think about it, is a little surprising, because the estimates of future payloads predict that more than 70% of future payloads are going to be composed of this synthetic consumable, meaning that they are going also to be composing the large portion of space waste. But we should not consider this as a burden, but rather a resource. And if we can use microorganisms to recycle and upcycle in this waste, we can tackle two problems with one. First, it helps minimizing the resupply of resources from Earth, but it also helps reducing space waste. And this in turn helps not only achieving a sustainable human presence in space, but if you want, also addressing ethical consideration on the production of waste in space. And this is what I'm focusing my research at the moment, funded by the Liverpool Trust at the moment at the University of Edinburgh, but as has been mentioned, I'm actually now moving to Cornell University, and this is going to be the main core of my research group. And here is just some very, very preliminary data that are really spin-off data from BioAsteroid, in which we have some starting indication that microgravity could negatively affect the capacity of the microorganisms to degrade plastics. So research is needed. But apart from all the research that is needed, I just wanted here to make a final point, a final wrapping up point, let's say. I do believe that synthetic waste recycling could really become a core and a promising tool toward a circular economy in space. This figure here comes from this perspective paper published last year, and we put waste recycling bioreactor on purpose in the middle to show how this could really feed other bioreactors for life support systems and in situ resource utilization. But this is really a circular economy, so it brings me to another point, which is how can an efficient circular economy in space pose benefit to Earth? We know, I don't have to convince this audience that space is really important for Earth. These are just some examples of technologies that have been developed originally for space, science, but now we use it in everyday life. This is something that I always like to say to the public when I talk about this. But not only ESA is very upfront on this perspective, but the United Nations do recognize the space activities as being key for the development of sustainable development goals. To make you an example, examples are later. Due to its limited resources, indeed, space, and due also to the stringent condition and the lack of resources, space does work as a model system, basically what we should be already doing on Earth in terms of circular economy. And just to give you some examples, so the 2 biotechnologies that I mentioned in this talk could feed on 4 of these SDGs. So this is just an example that I was trying to make before. So the take-home message for this is that, first of all, long-term and long-distance space exploration are going to require sustainable ways of obtaining resources in situ, but that microbial biotechnologies can really help on that. And not only they could help, but they can also help achieving a sustainable human space exploration, which in turn could also pose a lot of benefit for Earth, including supporting the United Nations SDGs. And this is a kind of a final pep talk that I wanted to give to the audience, which is that Europe is really leading, it's already leading all science regarding climate change and also the politics regarding climate change and environmental protection. And ESA is doing that very, very well, and our community is doing that very well. So yesterday there was a starting talk about who is leading which part of space science, and I make the question on purpose because I was thinking of this. I was thinking that we should really lead sustainable human space exploration. And the final point is that I would like to highlight that astrobiology and space biology could really bring a new vision to this topic. So to conclude, I would like to thank all the people that are involved in the work that I've shown you today, and you for your attention. I will be very happy to take your questions.
Thank you for your talk. Questions?
I spoke too fast.
I have one. A single well-prepared microorganism or a well-selected and adapted microbial community for doing this? What is your preferred approach?
Depends on the application, I would say. So for biorock, for biomining, for waste recycling, for what?
For biomining, for example? Well, for recycling. For recycling.
Okay. So again, I think it might depend on the application because I'm thinking of plastic waste recycling, or better, I should say, biodegradation. You might find wine microbes that work fine. Electronic waste recycling, the best case scenario is that you have a community, because electronic waste is going to be composed by metals plus plastic plus other materials. So if you have a community and maybe each one of them will degrade a thing, that will be great. But even in the case of you have one microbe that does everything, if you have a community of microorganisms and maybe each one degrades something, but then maybe you have some that have been engineered to take these molecules that have been degraded and then produce something else, so synthetic biology, that will be kind of key. So I think it's really difficult to have one single question. And I think if there is something that I learned from all this experiment is that when they tell me, for instance, okay, so does microgravity do something to microorganisms? The answer is it depends on 1,000 different things. And so, yeah, I'm sorry, it's not a good question answer, but is true. I really believe it depends.
Thank you.
I have one question. Thanks for the presentation. Really, really nice. I love the idea. So it is almost in the direction that you already answered. Synthetic biology for biomining. What are your after you do biorock, especially biorock, because it was ESA experiment? What would be the next step? Would you already go to synthetic biology, or you think we are not even there yet?
So one thing that is probably key is understanding if you scale up the system, is it still going to work? Because the bioreactor that we used was brilliant, was working fine, working okay for the perfectly designed for the cubic incubator, but it's 5 ml. So if you want to make it industrially usable, you have to understand what happens to a larger volume. And understand, okay, in 5 ml, we don't see much of a difference. If you have 1 liter, if you have 10 liters, probably it's going to change. And then I will probably see if other microorganisms could be used, if a community could be used, and if synthetic biology could be used to improve them. And in terms of improving, I think you can go 2 ways. One is improving their capacity to extract. And the second is improving their capacity to resist to the space conditions while they do, in this case, the biomining. So again, there are 1,000. I think we are really at the beginning of that, and which is exciting if you think that there is a lot of opportunities.
Thank you. Yeah, so now I think we can close. Thank you very much for all. We are running out of time, actually, but it was great, and see you all tomorrow. Thank you.